I'm going to introduce the speakers. They'll speak, and then we'll go to Q&A. So Julie works at the Association to Preserve Cape Cod as the Cape Cod Pond and Cyanobacteria Program Manager for APCC, and coordinates and manages their pond monitoring program and the cyanobacteria monitoring program. So she's got a huge job. We've got officially about 890 ponds on Cape Cod. So Julie and her staff have a big job. And really thankful that she's agreed to speak to all of us today. Liz Moran is a planning and restoration specialist at a consulting firm called Anchor QEA. She's an environmental scientist with a long career in lake and watershed management. And she's a member of the Cape Cod Commission team that is guiding the Freshwater Ponds Initiative. And so I'm really pleased that the two of them are here. Okay, Liz?
Okay, thank you, Ken, and welcome everybody. Thanks for gathering on what is apparently a very nice afternoon out on the Cape. I am beaming in from upstate New York, where it's also pretty nice, but I get to talk today about one of my favorite ecosystems, and that is the unique cattle ponds of Cape Cod. And so Julie and I are tag teaming this presentation, and I'm going to start off with some of the basics of limnology. So limnology is the study of fresh waters. It's kind of limnologists look at ponds in a very holistic point of view. So when we talk about the ponds of Cape Cod, we talk about the geology, the history, the chemistry, and of course the biology. So I'm going to do some of the basics, and then Julie is going to delve into much more detail on a lot of the biological aspects, which are, of course, really interesting, and talk about what do you monitor to figure out what's happening in your pond and is it healthy. So this initial slide here is kind of the geology. So looking way back, the kettle ponds were formed as the last glacier retreated maybe 14,000 plus years ago. You can see from this little cartoon on the bottom, chunks of ice broke off that retreating glacier. And that's kind of what we're reading about now, right? Is ice in the Arctic is also retreating. We're seeing ice flows. So when this ice sheet was retreating, some of these chunks that broke off got buried in sediment that was accumulating from the meltwater of these glaciers. And they stayed, eventually they melted, and they are of various, here's like an aerial photo of some of the ponds on Cape Cod, and you can see they vary in size and shape and location. And you also notice that there's not a lot of streams flowing in and out of these ponds on the Cape. And you probably observed that in your own travels in your neighborhoods. But these ponds have evolved over the years to have a real range of ecology based on where they are in the landscape, how deep they are. That turns out to be really important. And they provide really important and unique habitat for terrestrial and aquatic animals and plants who live there. Next slide. Here's another view of Cape Cod. This is a cartoon. A lot of the slides I'm going to use today, I have to give credit to the Cape Cod Commission as part of their Freshwater Initiative. They developed lots of these really good illustrations. This one is an illustration of the groundwater. So we know that Cape Cod, of course, is a sole source aquifer. It's where we get all of the drinking water on the Cape. But it's also divided into what we call lenses, which is where the groundwater is closer to the surface. So these are the significant lenses and their names. And you see that since we're focusing on Falmouth, the Falmouth ponds tend to be are in the Sagamore lens. And so there's interconnection then in these lenses, as well as connection of the groundwater ultimately making its way to the estuaries. So next slide. And ponds, when we think about the ponds, we think about them kind of from their geology and their history and their natural ecology, but in many ways we think about them as the way they provide what we call ecosystem services, which is how those unique ecosystems help support our well-being as people. And as part of the Cape Cod Commission's Freshwater Initiative, we did some initial kind of stakeholder conversations and talked to people about what do they value most in their ponds. And we got lots of ideas and thoughts and perspectives. Some of them are listed here. So not only are they good for, of course, habitat for aquatic plants and animals, but they're really important to us in terms of our sense of place, and then some of the mental and physical health amenities that they provide in recreation, aesthetics, just that sense of being with nature, environmental education. And then, of course, you've probably all been hearing for many years about the importance of ponds in the nitrogen cycle and how that they also impact the quality of our estuaries. So next slide. I said I was a limnologist, I think, at the beginning. And one of the key lessons that's in the actual, foundational textbook of limnology from 1957 is this quote, which is very important to, I think, when we talk about the ponds of the Cape. Ponds change. So you can read this quote that they seem to be permanent conditions of the landscape, but they're actually not. And they all will die. So you think, well, what do you mean they'll all die? And that means they will revert into some kind of vegetated wetland or some other type of landscape feature as they gradually fill in and become not visible as blue dots on this image. In fact, as the Cape Cod Commission does their Lake Atlas, and on a decadal scale, we're already seeing that, there may be several dozen fewer lakes each time they do these aerial images as they are turning in to wetlands. So next slide, please, Kim. So I'm going to just go through some basics. So the next couple of slides are going to be limnology basics. The first thing we're going to talk about is phosphorus, which is illustrated in this graphic. And you may think, well, why are we talking about phosphorus first up? And that is because phosphorus is really one of the key nutrients that supports the growth of aquatic plants and algae, and the primary producers, in the ponds that form the base of the aquatic food web. So ponds are essentially, in most cases, with few exceptions, are limited by phosphorus, which means that the amount of phosphorus in the pond is what determines the amount of biomass, like primary producers, bacteria, plants, algae, that form then food and carbohydrates that support the rest of the food web. So this is kind of just a simple schematic here. Phosphorus enters our ponds through groundwater. Some runoff, as we said, there are very few ponds that have defined outlets, streams going in, or rivers in atmospheric deposition. So when the phosphorus gets into the pond, it can be taken up for primary productivity by the photosynthetic algae, bacteria, and plants. And then that production forms the base of the aquatic food web, as you see here. So, and this is something that you'll hear a lot more about from Julie. The bacteria, the algae, the little suspended animals living in the ponds, the fish. And then you'll see here, there's some arrows that go down into sediment. And we're going to talk a little bit more about this in a couple later slides, because the interaction of phosphorus in the sediment with the phosphorus in the water column is one of the key determinants of how our different ponds respond. Okay, next slide. So, second limnology basic slide, and this is something that I'm sure you're all familiar with, and that is the temperature changes annually. As you start up here in the winter, even in the deeper ponds, the temperature of the water from top to bottom tends to be the same, right? And then when that temperature, well, there may or may not be ice, right? There's, in recent years, there has been much less ice cover on the Cape Cod ponds. But when the surface of the water is ice-free and the temperature is about the same from top to bottom, there's no density stratification. You know, cold water is denser than warmer water, right? So the winds are able to mix those ponds from top to bottom. And then as the summer progresses, the upper water starts to warm and that density gradient starts to form. So you have a warmer water layer floating on top of the colder water that continues through the summer. And then in the fall, eventually the ponds start losing their heat. That thermal stratification eventually becomes weak enough that the winds can blow it and mix it from top to bottom again. So there's a lot of information on this graph, and I want you to remember this when Julie starts to talk about the enhanced monitoring that's going on the Cape Cod ponds now, because in the spring and in the fall is when you get a full sense of, if you're worried about something like phosphorus, how much is there really in that water column? So that's as opposed to in the summer, you know, so, okay. So next slide.
Okay, Liz, you've used
Yes.
the water column a couple of times. Can you say what a
Oh,
water
yes,
column is?
thank you. Thank you, Kim. The water column is just the gradient from the surface to the bottom. So the water column is if you put a cylinder through the middle of your pond, this would be, and pulled it out, that would be the water column. And as you can see from this slide in particular, water temperature tends to be different seasonally through the water column, so warm in the top. And this slide, thank you, shows that kind of the interaction of that stratification and some of the important chemical and biological processes that are going on in your pond. So this would be a picture in the summertime, right? This might actually be what many of the deeper ponds on the Cape look like right today, if you went out and did a sampling event. So you have this upper layer where the sunlight can penetrate, and that's where you get primary production. So you get the phytoplankton, which is, you know, algae and photosynthetic bacteria, tiny organisms suspended in the upper waters. They are capturing that sunlight energy through photosynthesis, right, and making carbohydrates. So they produce oxygen in that process. They also, of course, use it, and Julie will talk more about this as well. The upper waters tend to be subject to turbulence. We see that depending on how much wind there is, or I saw a comment in the chat, maybe how much wakeboard activity there is in your ponds as well. That can translate into turbulence. Then you have this layer where the temperature changes really quickly, and then you have a deeper water layer. Now, for some of the deeper ponds, this deeper water layer can remain isolated from mixing, and also there's no source of photosynthesis going on because it's below where the light can penetrate. So there's no addition of oxygen that goes on in the summertime in these deep waters. but there is still respiration going on. So the animals and plants that are up here in the water column, as when they die, they will settle down to the bottom, and then they will be decomposed in a process that uses up oxygen. So sometimes when you look at some of the data for your pond, you might say that there's low concentration of oxygen in the deep waters during the summer, and that's why. And it's all kind of this supply and demand question, right? So shallow ponds, they don't have a very big volume of deep water that remains isolated from the atmosphere, or the light can penetrate far enough that photosynthesis, either from rooted aquatic plants or from these suspended plankton, can actually produce oxygen. So let's move on. So we've talked a lot about phosphorus, and one thing that we wanted to illustrate with this is that because these ponds are largely fed by groundwater, you know, we've said several times they don't have streams and rivers running into them. They're connected to these water lenses that we saw on the slide. So phosphorus on the surface can make its way down through the generally sandy, gravelly soils, and make its way into the ponds. Same with nitrogen. Now, look at the difference in the size of the arrows, the green arrows for phosphorus versus the orange arrows for nitrogen. Phosphorus gets held up in the soil profile. There are chemical reactions that occur. And so you see these arrows getting smaller. Where nitrogen, those arrows are the same. Nitrogen is soluble and it moves through the soil profiles. So that's not to say that there's an infinite capacity of soils to bind phosphorus. And in fact, when Falmouth was looking to expand their wastewater treatment infiltration beds, they had to look at some analysis of, okay, what's the nature of the soils down gradient? What direction is the water going to flow? And how many years of capacity do you think we have in that soil prism, call it, that area of soil that's going to be subjected to the infiltrating wastewater? How many years of capacity for phosphorus binding do we have? So there's a lot of research and analysis and environmental impact assessments that go on because of the vulnerability of ponds to phosphorus. You know, we don't have a lot of farms the way we have where I can, you know, live up here in New York State. You know, we have a lot more big ag. We don't have that. But septic system discharge is a big question. So that's that. So the takeaway lesson is the landscape, what's happening on your landscape in your watershed, which is the area of land that contributes that groundwater to the ponds ultimately, that affects nutrient flux, which then affects water quality. Okay, next slide. And another key concept, the nature of the ponds themselves affect how they respond to those incoming nutrients. So this is another technical term, morphology. It means, or sorry, morphometry. The depth, what's the size and shape of the ponds? How long does the water stay in the pond, right? Because water's seeping in through the groundwater, and eventually, you know, the pond levels certainly fluctuate because the groundwater table itself is fluctuating. You know, wet years, the pond levels are higher. Dry years, the pond levels are lower. But eventually, the water seeps through these ponds. And how quickly that takes also gives us a sense of how long those nutrients are going to stay in the pond. And also, if you do something on the landscape to try to minimize import of phosphorus and nitrogen to the ponds, how long is it going to take before you see the difference, right? Because that water has to slowly seep in there. And I think there's another good example of that in Falmouth with Ashumet Pond, where, you know, it took many years for a plume to get from the mass military conservation, is that the right name for it?
Yeah.
To actually reach the pond. So again, residence time is key, connectivity is key, and then also there's differences in the pond response based on their ecology. So what's the fish community? Do they have an invasive species? And then management. What have people been doing? Is there a constructed herring run connected? Have there been fish stocking exercises? So all that can affect the pond response. Okay, next slide.
Okay, and Liz, connectivity?
Connectivity. How are they connected to their neighboring ponds within their watershed lens or within their groundwater lens, right?
Got it.
So some of them are going to be, ultimately everything's flowing towards the oceans, but some of them are going to be upstream, some others are going to be downstream. So what else is coming in and how connected are they? That's what connectivity is.
Thank you.
Okay, I'm passing
All
the
right.
baton to Julie.
Right. So I'm taking as they say, a deep dive into the water. And so just looking at what the water is composed of, you're looking out over your beautiful pond, and it's composed of dissolved substances and suspended particles. And so the dissolved substances that you don't generally see are the gases, minerals, and the organic compounds. And the suspended sediments particles are either the sediments, like the silt and the clay, or the microscopic living organisms, which is what you see when it turns green because of the phytoplankton. Those are the cells that are photosynthesizing, or the zooplankton, which are the little animals, little shrimp-like things, copepods, that are eating the phytoplankton. And so on the left, you can see measures of pond health. And so I'm going to, in the next couple slides, I'll describe more about why those things are important. The temperature, oxygen, the pH, the nutrients of nitrogen and phosphorus, the water clarity, I'll say something more here, and the chlorophyll, which is a way of accounting for the plants that are in the water. But the water clarity, we have a simple technique that we can use called the Secchi disc, and I'll show you one later. But what it's accounting for is those suspended particles. And the non-living ones, the clay and the silt, would be things that you would see a lot of after a storm event, if you had areas around the pond that were eroding. And the living organisms would be the plankton and the blooms that we'll see. So the water clarity is an important component contributing to the health of the pond. If it's not very clear, that's not a very good thing. Next. So one of the things that is considered not so good is eutrophication, fancy word. So what's eutrophication? Eutrophication is refers to the process of enrichment of the water body with nutrients, particularly nitrogen and phosphorus, and the impacts of that enrichment. The enrichment will cause algal blooms, and if you have enough of that, you have the decrease in the oxygen depleted from the water. And I'm going to explain that in the next slide. But here you can show that the fertilizers running off are part of, the nutrients coming in. As Liz has already shown you, the algae blooms occur because of the nutrients being present, as well as the sunlight. And then the bacteria at the bottom of the pond use the oxygen to break down the algae, and that's decay, and that's another source using the oxygen. And then if you have too much decay and too much use of oxygen, then the fish will suffocate. So there's a lot of the health of the pond revolves around how much oxygen is in the pond. So fertilizer, most of you have used that at one point or another. That's the N, nitrogen, P, phosphorus, and K usually, which is the potassium. The runoff, you understand that term, is just what's coming off the land. Algal blooms or cyanobacteria, which used to be called blue-green algae, those blooms occur as a result of these nutrients. Bacteria decay, that's generally speaking at the bottom of the pond, where they're using the organic matter there and using oxygen to break it down and then put the organic compounds into the water again. And water column, which Kim asked Liz to is from the surface of the water down to the bottom. And we typically will see a difference, especially during the day, of the amount of oxygen that's being produced at the surface and depleted as you go down further in the water column. Next. So the thing is, you could think of your pond as being somewhat static, but it's changing all day long. The photosynthesis takes place in which in this case, algae or cyanobacteria are using carbon dioxide and sunlight, and they're making the carbohydrates, and they are giving off oxygen. Yay, we're all grateful for that. Respiration, we breathe, the fish breathe, the zooplankton breathe, and the plants breathe. The plants respire also at night, and they use their carbohydrate. What are we doing with these carbohydrates? We get energy from it. We get energy from our food, and what they do in the water is that the plants divide and make more cells. So they're using the energy, using respiration and using up the oxygen and giving off carbon dioxide. And that also happens at the bottom of the pond, where they're using the oxygen to break apart the carbohydrates and the other organic matter. And then you have organic matter in the water, but you've lost the oxygen. I hope that's clear. Next one. This just illustrates what I just said, but in day and night. So during the day, the plants use the carbon dioxide and they produce oxygen, but it raises the pH because the whole process of photosynthesis is it's using carbon dioxide, two oxygens, kick those off, more oxygen. It splits water, H2O, again, more water, more oxygen. But at night, when they respire, they are using the oxygen to break down the carbohydrates. So that's like day and night. And so it also, down below, you can see the line where it says the water temperature. That's easy to understand. It goes up when the sun is out and it goes down at night. You can see that the dissolved oxygen goes up and then it goes down at night. And so if you wanted to look at the health of your pond in terms of oxygen, which is important, the best time to take that measurement would be very first thing in the morning before the lights come on. Next. So what are cyanobacteria and what role do they play? Well, we've already said that they're photosynthetic bacteria that live in the water. There are over 1,000 species, and they're not all toxic. The illustration on the right of spirulina or spirulina, depending upon what you want to call it, they're actually selling it in the health food stores for having the phycocyanin, which is what we talk about when we're measuring the cyanobacteria in the water. And it also talks about chlorophyll and how healthy it is. So that's one example of a blue-green algae or a cyanobacteria that's not toxic. But the cyanobacteria that have been around for 2 to 3 billion years, they have developed quite a few strategies of success. And one of them is called buoyancy, or how buoyant. They can adjust their place in the water column, again, from going up and down in the water, because they make little gas vesicles or air bubbles that allow them to go up and down. They can produce toxins, which you may have heard about. I can say something more about that. They have the ability to use nitrogen. So if nitrogen was limiting, they can produce it. So then they just need the phosphorus. And Liz has shared this bloom triggers picture here, where what you'll hear is these algal blooms, these cyanobacteria blooms, require nutrients, warm temperatures, and calm waters. And as we reflect a little bit on the occurrence of the cyanobacteria blooms here on Cape Cod, they aren't as bad now as they were during the drought. And I'm reminded of that in the last 2 years when we've had cooler summers with quite a number of rains. I think that these bloom triggers were higher in the past. Next. This slide is an example of the 4 most common cyanobacteria that we're finding in our samples. I'll start in the top left and work counterclockwise. The microcystis in the top left is one. Those little dots are each cells, and from that you can get the toxin microcystin. And that's the only toxin we've been able to analyze so far on the Cape in a regular way. And the Barnstable County Lab has been very generous for 2 years and did those analyses for free. And now they're charging, but they worked out their techniques and we're very grateful. So then you can have an association in how much toxin you get from how much of the biomass of this microcystis. The one below that is called a phanoxamenon. It can also has some toxins. Many of these have genes that can produce more than one toxin depending upon conditions. Oricinia is the next one over to the right, and up at the top right is Dolichospermum, which is the most common, and you're seeing it in this slide in two forms. It's got a curly form and a straight form. The straight form is in the center of the slide, and I want to point out three characteristics which are part of its advantage. It's a string of cells that you're looking at, and each of those cells has little sparkly areas that are where the gas bubbles are, and that's part of its buoyancy. The big round circles in the center have cells with hard shells on them, and those are the quote-unquote seeds. They're called aconites, but they're seeds. So when your bloom dies, those aconite seeds are still there. They drop to the bottom, and they can make more cyanobacteria blooms whenever the conditions are right. And in the middle, briefly, that's a heterocyst, and that's the cell that can fix the nitrogen. And by fixing the nitrogen, I mean it can absorb the nitrogen out of the water and provide it for the cells. Next. So this is my favorite slide, I will say. This is describing what happens during a cyanobacteria bloom. Initially, you have the little dots. See where the red is? All those little dots? That would be the plankton, the cyanobacteria cells in the water column. Well, then they use their gas vesicles and they go up to the surface. At the surface, they get more light and they can produce more quickly. But then the wind comes along and it takes those cells that have buoyed up to the surface and it blows it over to the edge of the pond, depending upon the wind direction. And that is what we at APCC and the volunteers that work with us collect and look for, these what we call scums. Terrible term, but when they show up on the shore, that's how they get there. And it's not that the whole pond is as dense as what you see on the shore, but it is a concentrated amount. And I feel very justified in using that concentrated amount because that beach is where the young children are. That's where the dogs are. And that's where they're going to come in contact with cyanobacteria. And so we go to the edge, we collect that as a special sample, and then we sample in the water to see how much cyanobacteria is there. Next. So why are cyanobacteria a problem? One, they're increasingly abundant because the ideal conditions by which they can multiply rapidly is warm water, nutrient-rich water, and calm waters. And those conditions are becoming more common. They are potentially toxic, and that includes two things in the water, so that when the zooplankton are feeding, if they don't take in the cyanobacteria, like the zebra mussels in Lake Erie, they can spit it back out again and then consume the rest of the other good plankton. And so then you leave the cyanobacteria in the water and they have all the nutrients for themselves. It's not quite as simple as that, but in general. So that's one part. It's a problematic because those toxins are making the waters unpleasant for our skin irritation, and they can have ill health effects, especially if it's drinking water, but also for your pets. And so the range of effects will depend upon the person, how sensitive you are, and how much contact you've had. They also degrade the habitat. It looks terrible, smelly, ugly, and property values go down when you have a lot of cyanobacteria blooms. Next. So how healthy are the Cape Cod ponds and lakes? What APCC has developed is something they call the state of the waters. And it's important, this is a map that came out from last year. It was in our annual state of the waters. It says 2023, but there's no 2023 data on there because it takes us a year to accumulate all the data. So at the bottom, you'll note that it's a data average of 3 out of the 5 most recent years, going back from 2022. So this year's report, which if you want to hear the results, you can come this Sunday to the APCC annual meeting, which will be in Dennis, and you can look it up. We're glad to have anyone come. And it's just, we're not finished with the report. We just have the very beginning of those results. So how are these ponds graded? Well, one is using something called the Carlson Trophic Index. Bob Carlson created this back in 1976. I was fortunate to work with him in Ohio, and it involves 3 things. Total phosphorus, as Liz mentioned, is very important. Chlorophyll, which is the result of the plankton absorbing the light and the phosphorus and making carbohydrates, and a Secchi disk depth. And I mentioned that earlier, but the Secchi disk depth is how far you can see into the water using, I'll show you in the next slide, the Secchi disk. And if you cannot see 4 feet into the water is considered eutrophic. And eutrophic, I've already described, is essentially going to mean you're going to be oxygen depleted. You're going in the direction that you're going to be losing oxygen, and it will not be healthy. So that's one measure. Putting those 3 together gives you a score in the Carlson Trophic Index, which is what we use as to how healthy the ponds are. And the second method was using the cyanobacteria monitoring, and when ponds were considered had sufficient bloom, and this is using Massachusetts Department of Health view of the visual bloom. Also, if we had microcystin data that was greater than 8 parts per billion, it would be considered a public health advisory. If any of the ponds get a public health advisory, that is would consider that pond to be unacceptable and need of restoration. So to get the data to make the state of the waters, we're very fortunate that the Cape Cod Commission and Barnstable County that's funding it funded these freshwater pond monitoring program, which is monitoring 50 ponds. We chose them to represent Cape Cod in all 15 towns. And there's the Secchi disk on the right-hand side. That's what it looks like. And as you lower that into the water to where you can't see it and then bring it up to where you can see it, that's how you define the Secchi depth. Okay, that's how you get there with your eyes. One could have a whole session on seeing the Secchi disk. You could see it with a tube, without a tube, and under whether it's shaded or with there's a lot to be discussed there. But in addition, our team goes out and uses a data sonde and collects every meter in the water column. You all know what that is now. So every meter going down to the bottom, we get measures of the oxygen and pH, salinity, and the ortho. Let's see, help me here, and the conductivity. And the temperature, of course. So then we also collect at 4 different depths, if it's a deep pond, the bottles that go up to the Center for Coastal Studies where they get analyzed. I saw a question come in earlier that asked about the time of year when the ponds turn over. And last October, it turned out only half of these ponds had turned over. And it was a very key, important aspect to understanding, as Liz pointed out, the overall water chemistry and the water quality. So we extended the program. Instead of being from April through October, we now monitor from March all the way through November. And the ones we monitor in November are the ones that have not turned over. So we can't get to all of them. It's very cold. And we have to wear dry suits when it's really cold. And we don't take volunteers in those months because it might be not so safe. But we get that data. So that's collects the information you need for the trophic index, among other things. Next.
Julie, can you just say what conductivity is?
Conductivity. That is the conductance in the water in which in this case, it's also referring somewhat to the salinity. So how many ions are in the water? Yeah, so you have plain H2O, and then you have a lot of other constituents which allow the water to be more conductive, if you will, if you think of it in terms of electricity, than this plain. That's my best for right now. I don't have it written out. So the second grading system that I mentioned for the health of the ponds was the cyanobacteria risk. And this is an example from every year at the end of the year when we made all these analyses throughout the year. We have for each pond the record of whether it was acceptable, whether it was a potential for concern, meaning a scum was found and photographs were sent and the amount of biomass was looked at, and it either was considered a pond that needed a public health advisory or not. So if it was present in some numbers, could be considered a potential for concern, keep your eye on it, monitor weekly, or it's closed. So you can see that Ashuma Pond in 2023, last year, many of you may remember, it became closed, it became public concern in early September, and it just never let up. It just was never acceptable. The bloom continued, didn't just come and go. Whereas Coonamessett Pond, it had a couple of potential for concern, and it's a little concerning to those of us that work with it because it can turn green, but it doesn't end up with scums on the side. So we don't have a basis for moving it through the process, and we will be working on that again this next year. In Crooked Pond, you had some blooms in the 2021 years where it was a public concern, but it has not had that happen since then. The main point of this slide is, one, it's not predictable. Here are these ponds. You know, there's, can you predict the seasonality here? Can you predict for the same pond? Or it's just really hard to get the conditions to where you could predict what's going to happen with these blooms. It can happen when the conditions are right. And on the right, you will see West Reservoir. This is your classic picture. This was taken with a drone on August 1st last year by Ole Becker. Some of you may have seen Save Our Ponds, which is a video series put together by Woods Hole. And Ole Becker was the one who did the wonderful work on that and interviewed people for it. On the right is a cyanobacteria bloom, West Res, which is blooming right. Well, it just stopped this past week. So it's just coming out of having had a public advisory on it. And so this is my last slide in the series, which repeats what we just did with Kim asked about. So the acceptable is blue, the potential for concern is yellow, and the use restrictions are red. So this was basically last week. And you could see out of all the ponds, they're mostly all blue. The ones in Falmouth that are yellow were potentials, and all of those cleared out. It was just a scum 1 week, and then it was gone. So those would all be blue now. The red in the middle is Lake Wequaquet, for which there's a lot of questions right now. It's a big pond. Scum's showing up just at the yacht club, but it's also throughout the pond in different areas. And then the ones in Brewster, those have cleared out, so that would also be blue. So if you think about it, I mean, we're just monitoring 134 ponds at 150 sites. That's not bad. You know, Cape Cod's actually looking pretty good. It doesn't account for everything with the health of the pond. But given that it's August, and this would be the warmest time of year, and potentially the greatest potential for these blooms to occur, this year in 2024, Cape Cod is looking pretty good for all our concerns. And it doesn't mean that the nutrients aren't there and that it's not eutrophic or that it couldn't happen anytime. But we should be grateful for how it is looking right now. Over to you, Liz.
In addition to the great work that is being done now through the Freshwater Initiative and lots of other partners, we are getting, lots of data and information on the ponds. Fortunately, on the Cape, there's been a longstanding program, the PALS program, Pond and Lake Stewards, which has collected snapshots of pond water quality, typically this time of the year. So late summer, when conditions are the ponds are at their most stratified, they're at their highest risk, typically for algal blooms and cyanobacterial blooms. So this graph here that you see is the surface water temperature of ponds over time. And red means that there's an increasing trend, which means the ponds are getting warmer. That probably doesn't surprise many of you because, of course, the air temperature is getting warmer as well. So ponds that are filled in are getting, the circles are full. They're getting warmer at a statistically significant higher rate than the ponds that are open circles. The blue ones are not showing a trend at this point. So the next slide shows the same data set from PALS for phosphorus. And this shows us that approximately half of the monitored ponds on the Cape show a statistically significant increase in total phosphorus concentration. So as we talked about, phosphorus is limiting to the growth of aquatic plants and algae and cyanobacteria. So the fact that that is increasing is also potentially of concern. Why is this increasing? Well, there's a lot of development that's been going on the Cape over the past few decades. There's more people, there's more wastewater, and there's more impervious surfaces. So there's less opportunity for water to filter through the ground before it reaches the pond. So that's just, something to understand because, we started off with the Limnology 101 that ponds are kind of ephemeral in the landscape on geologic time. And then we talked about eutrophication and that process. And then we looked at some data. So we see that these are actually coming to pass slowly, but surely on Cape Cod. So my last slide is kind of key takeaway points, right? This is Ponds 101. So here's what we hope that you all can go home with and share with your community and family. Ponds are changing, and things that we do affect the rate of that change. We talked a little bit about how unique kettle ponds are in, their lack of inlets and outlets, the fact that they're just windows into these interconnected groundwater lenses, essentially, that phosphorus is a really key factor in pond aging and pond health, but it's not the only factor affecting rate of change. Temperature is certainly critical. As the water's warm, it affects the pond structure, right? The thermal structure, those were those diagrams that we looked at with the layers of cold water and warm water. That happens, right? If you have a really warm fall or a really warm spring, you're going to see differences in stratification. Like it took until November, Julie said, for some of the ponds to undergo their thermal mix, their fall mixing period. And I think all in all, this points to the real importance and value of long-term monitoring. So with that, I'll turn it back to Kim.
Great. Thank you. Thank you very much, Liz and Julie. Lucinda, do you want to go into the chat questions? And
sure. Our first question was for Julie, and that was, is inflated bladderwort, a sign of a healthy pond?
Well, the bladderwort, also known as utricularia, it's an interesting one. The plant that we see here most common on the Cape is showing up in eutrophic ponds, in shallow ponds. As a group, these bladderworts can live in leaves in the tropics, and what they are is a carnivorous plant. They take in animals and use that as their protein source, so they can get all the nutrients they need by using their bladders, if you will. But this particular one on Cape Cod it's eutrophic, so it's not an indication of low nutrients, which it could be, another species in another place. I hope that helps. But Utricularia.
Thank you. Another question was, at Uncle Harvey's Pond in Orleans, I find floating debris accumulates. Compost on the surface and often starts a small cyano bloom. Would the removal of dead filamentous green algae and dead water lily leaves reduce this possibility?
I could take that one. Well, the floating mats could be benthic cyanobacteria or not. And yes, if they're on the surface, they are going to be decomposing, so they will be using oxygen, which is not going to help the fish in the pond by using up the oxygen. So if you have a place near your pond where you want to, have a small compost pile, that would be perfectly right. But don't go taking out your rooted vegetables. You need to consult with your conservation agent for your pond if you have a problem with the rooted vegetation, because they're very strict about that, and they'll give you a permit. Like if you pond that's completely surrounded by rooted plants, you can get permission to have an access for your boats if you want. They'll allow you to do that. But this is my opportunity. Sorry, I'm going to just add on that if you take out the rooted plants, they are using the nutrients. And if you remove them, you're going to have the nutrients still there, and you're more likely to get a phytoplankton or cyanobacteria bloom. They're doing you a favor. They're just like the trees in the forest that are harvesting the carbon dioxide and sticking it in the ground in their wood. These rooted aquatic plants are harvesting and keeping some of those nutrients that you have too much of in the pond.
Thank you. Liz, you had answered part of this about when does the fall turnover occur, but they also wanted to know what are the signals of this?
Okay, so in the fall, this pond starts to lose heat to the atmosphere as it gets cooler. You can see the fog on the surface of the lake in the morning. And that's a sign. So eventually the water cools, and what happens is, as the water cools, the density difference between the layers gets smaller. And that means it takes less wind energy to actually mix the water. So that's what happens. The signs of that, really, the best way to do it is to look at temperature, measure temperature through the water column. So from the top of the lake or pond to the bottom. And that's what the APCC science team is out doing in the fall to figure out when these ponds are mixing. So did that answer the question?
I believe so. Thank
Okay.
you.
Sure.
Liz, Catherine wanted to know whether the wake of boats would affect the cycle of thermal stratification.
That's a really good question. Yes, certainly. Depends on, again, the temperature gradient between the top and the bottom. How deep that warm upper layer is. If you have a high energy propeller mixing that in, it could start to disrupt that stratification. But again, if you're in a deep pond, it's unlikely that the propeller action would be that intense. But I don't know. I think that's a good kind of physics question, like how big of a pond and how fast of a boat, and what is the thermal density gradient when you're out boating. But yes, it certainly does mix those waters, and that can help distribute nutrients through the water column, right? Because if you get the deeper water mixing up into the upper sunlit area, any phosphorus that's in that deep water that gets up into where the photosynthetic zone can occur, that can help spur the, algal blooms and cyanobacterial blooms.
Thank you. Another question for you, Liz, was why does the temperature gradient prohibit mixing?
Okay, that's the density difference in water is what's actually prohibiting the mixing. So colder water is more dense. We all know that, if you put ice in a glass, the ice floats, right? Cold is at the bottom, warmer is at the top until it freezes. So that density gradient is strong enough that unless there's, a hurricane force wind in a very unprotected water surface, it's unlikely to have enough potential energy to mix the water layer.
Thank you. I have a couple more questions for you, Liz. The next one was from Peter and Karen, and they wanted to know what is the pond depth required to maintain temperature stratification and turnover behaviors?
Oh, interesting. That's something we've been working with as we've been looking through all the historic data for the Cape Cod Freshwater Initiative. And the cutoff point seems to be somewhere around 9 meters before you can get like a stable deep layer. It could be a very small volume of cold water. But if your pond is about, somewhere in that neighborhood, 9-10 meters deep at its deepest point, then it seems to be likely to develop thermal stratification. Again, it depends how big the pond is. It depends on how well protected it is in terms of wind fetch and such, the surface area. But that rule of thumb seemed to work as we were looking over data from, several hundred ponds.
Thank you. And the last question in this section, Liz, is my pond is 11 meters deep at its deepest point. Is that considered shallow or deep?
I would consider that right on the range of deep.
Okay.
I mean,
I can
that's
add.
like the shallowest of the deep ponds. Julie, what would you say? It's kind of a judgment
Well,
call.
all I was going to add was that when we had all the data on the ponds, trying to decide the 50 ponds on Cape Cod, it was 12 feet is what they use as a shallow pond. I mean, people can, but that was the definition that we used was 12 feet shallow. Everything above that was deep. And obviously, 15
That's not
feet.
the same as, right,
No.
right. That's not
So
the
it
same as
was-
will it develop a stable layering system though.
No, and actually all of the ponds, with maybe the exception of one, did have thermal stratification.
Mhm.
All of them, all the shallow ones as well as the deep ones. Yeah, at some point in the year.
Thank you so much. So these questions are for Julie. And Ellen wanted to know what is the effect of cyanobacteria on aquatic and terrestrial animals?
Well, there's the study that was done on the herringfish, which showed that the cyanobacteria toxins did show up in the flesh of the herringfish, and that was off of Brewster. The cyanobacteria that are toxic, there are the reports, sadly, of some dogs have died. Some dogs are taken to the vet and treated. So there's those kind of animals. Cyanobacteria are part of the photosynthesis in the pond. So if they're not toxic, they could be food for some of the animals and not be a problem. And so, but the bloom itself, if it's a bloom, would be one, produce using up a lot of oxygen at night and therefore not a good health signal for the pond.
Thank you. Peter and Karen wanted to know what constitutes restoration after a public health advisory. All that they've been able to see is wait and see and resample.
What's listed on the Massachusetts Department of Health website is they call it rescinding, that is taking down the advisory. And the way that works is that you're supposed to have 2 weekly samples in which the bloom is not present. So if you have a bloom and then it appears to have gone away, you don't rescind it right away because the cells break down. All the toxins that were in the cells are now in the water. And this is the hardest concept for most people to understand because they say I don't see it anymore, therefore it's not there. It's not a problem. But there is an extended time after it's there where when the cells die, they release the toxins into the water. And so the quickest, once it is given a public health advisory, would be in 2 weeks, 1 week in the middle, would be about 10 days to rescind the warning. Yeah.
Thank you. Another question is from Betsy. How large by square footage does a cyanobloom have to be to trigger closure? Is it by the ratio of the total acreage of the pond?
Well, that's a good question. I think you need to ask that question to the Massachusetts Department of Health.
Okay.
They get the photos, they make the decisions. I've never seen their book of how they make the decisions, but it's really, really difficult because some of these cyanobacteria are growing on the bottom. Some of them, like I showed earlier in the slide, they float up to the top and then they're very evident. And so if you want to go in square areas, just being able to even try to count, and we'll have a drone out there later this year if some blooms show up. Even if you could capture it with a drone, we're also working with satellites, and they're nationally working with satellites that have the sensitivity to pick up the pigments. And so that's another way we can, as we go forward, because we have a project in the SNEP project, where they're looking at all the Cape Cod ponds, and they will be able to look at the pond from the satellite and be able to read using the Secchi data and the chlorophyll data that we've given them. They'll be able to judge how much is there. But yeah, it's really hard to answer that in terms of square meters. Or Lake Erie once had the bloom 17 miles.
Okay. Peter and Karen were wondering if your pond is downstream from another with a potential for concern. Do we have an increased risk of a problem?
I would generally say no. I mean, whatever's going on with the connectivity would be underground, and that would be some time before whatever was in the groundwater, in the groundwater lens, would reach from one pond to the next. Having said that, it is another good research topic that some of these ponds have microcystis as the dominant, and other ponds in a cluster have the Dolichospermum as the dominant. And I don't think we know enough about the ecology of those two or any of the others actually to predict when and why you're going to have one kind of cyanobacteria versus another. Yeah, so I would say no, I wouldn't be worried.
Okay. Jackie wanted to know why to measure pH and conductivity. I believe this was done during Liz's section.
Well, the pH part, I will say what's the concern with pH is largely at the low end, which is when the pond gets more acidic. And that happened with the acid rain, which is what drove the National Seashore to create this project of monitoring ponds, which has been going on for over 40 years. And fortunately, because we've reduced the amount of smokestacks and acid rain that we had, they've been able to see those ponds increase. What I remember from way back when I was studying this, the fish, they have, you know how fish are slimy? Well, that slime is helping to protect them from the bacteria. And if you get low enough in acidity, you are removing the protective slime that they have around them. And there's that, and then there's how much oxygen. You know, a trout needs to have cold oxygenated water. Well, some fishes are far more sensitive to than others, but pH is really important in terms of the oxygen that's available, and a lot of the organisms that live in the water. So that's the answer to the pH part. On the conductance, again, I refer back to salinity, conductance. Some plants and animals are going to be happier in salt water than they are in fresh water. And so having an idea about the amount of conductance will affect the community. That's a simple explanation. There's probably a lot more to it.
Thank you. Ken wanted to know, what is the relationship between the thermal stratification event and the algae blooms or eutrophication?
Well, Lynn, I'll send it over to Liz to say more, but just in terms of the algal blooms that we see sometimes on the Cape in the fall, is that you've had, as Liz said, you're mixed up. You've got the things that were on the bottom are now mixing in the water. Well, what is that? Well, that's the nutrients. So now you have more nutrients available once you've mixed than you would have had when it was all stratified. Liz, what else do you have? You have some more to say about
I
that.
would add to that, there's another complexity that we didn't really get into too much. And I don't know if Kim can switch back to maybe slide 8. So quite a ways back. It's that picture of the phosphorus cycle.
Right
Yeah,
there.
that's it.
Yeah, this one. So when the if you think about this deep water, like overlying the sediment, as oxygen gets used up by decomposing all the, the growth that's happening in the upper sunlit layer, phosphorus that's in the biomass, the plants and animals that are being decomposed, that phosphorus gets released. Now, some of it could be, depending on the nature of the sediment, if there's a lot of iron, for example, minerals in that sediment, and there's oxygen and the pH is within a certain range, that phosphorus can get held in the sediments, right? It gets put into chemical complexes with that iron. But if you start to run out of oxygen, the chemical reactions then that happen in the deep water right at the sediment surface can release that phosphorus back into the water column. And that is kind of a complicated answer to the question of how does thermal stratification relate to eutrophication? Because if that phosphorus gets released and not held in the sediment, then eventually, as the pond mixes, that can be brought back up into the upper sunlit layers and stimulate more production, growth of algae and cyanobacteria. And I know I saw one question come into the chat about restoration techniques and alum, and that's not part of our Ponds 101, but one remedial measure is to put alum compounds in the deep water overlying the sediment, because that complex between aluminum compounds and phosphorus is stable even when you run out of oxygen, and it's stable over a broad range of pH. So that's why it is a remedial measure that has been employed on some of the ponds. So not to get into Ponds 102, but that's
Stay tuned.
Stay tuned.
Susie had asked, she said that she knew that Julie had mentioned that there might be less cyano because of the rainfall, but she wondered if there was another reason.
Well, the temperatures in the ponds haven't gone above into the 80s much this summer. I mean, it's mostly in the 70s at the highest. And so temperature is a really important factor in the bloom. So you can have it show up, and if the temperatures aren't warm enough, and again, it's probably species dependent or genera dependent how much. So that's a piece of it. There's the temperature.
All right, thank you. The next question was, would there be a benefit if we remove some plants and weeds or growth in the lakes? If so, what is the best option to help clean them up? That's from Grant.
Help
Well, Julie
clean.
started to answer that, right? With is it helpful to take some of the biomass out of the pond? And you said yes, if you have a compost area.
Well, I know that the rules in these ponds, as far as Cape Cod is concerned, is the conservation departments would need to review it. I had a different situation with the Mystic River, in which the invasive plant was the water chestnut, and no one cared. They really wanted to get rid of that for all kinds of reasons, and there was no restriction. So we were able to take tons, literally tons away. And when we did that, we were removing nutrients. So it was really a great thing for that river. We can't apply that here exactly on the Cape. And I warned, because I warned even at Mystic River, when they took them out with the machine, and it wasn't just that one, it was another of the aquatic weeds. Right after they took them out, we got a cyano bloom. So you have to pick your flavor of which kind of plants once the nutrients are there. But the good news about the water chestnut was we could actually remove the nutrients, because the nutrients are in the plant. And that would have been the case for Cedar Lake with the fanwort too, if you could get it out of there, because otherwise it's just going to die and be on the bottom of the pond decomposing.
Thank you. Maggie wanted to know, further to the bladderwort question, would masses of decaying bladderwort be a likely contributor to an algae bloom?
I don't know about the algae bloom. It's going to reduce the amount of oxygen. As they're decomposed, there's going to be more organic in the water, which I presume the plants could take up the nutrients that then were released through the decomposition. So that's where my mind would go with that.
All right, Josephine would like to know, what are the most impactful actions that we can take to regenerate the health of our ponds? Is it replacing septic systems?
Well, at APCC, we generally say, whatever you can do to reduce the amount of nutrients to go into the ponds, that's the number one thing. And every single person who lives by a pond that's fertilizing their lawn, that means please don't fertilize your lawn. Septic system's much more expensive. And I'm relatively new to APCC, but I will say I'm very proud of the fact that Andrew, years ago, pushed for funding at the state level to help our towns, so that when septics needed to be replaced, there was funding at the town to help the owners. Because otherwise, oh yes, you should, replace your septic system. It's just going to cost you $20,000-$30,000. Most people don't have that kind of just change to throw. So anyway, whatever you can do to reduce the nutrients is what I would put as the number one thing you could do.
Hampton has a question, and I'm going to apologize if I don't pronounce the words correctly. It says, why is the metalimnion usually more oxygen rich than the epilimnion and the hypolimnion?
Okay. Switch back. Can you go back to one more? No, must be ahead one more slide. Okay, yeah, this one, no, you had it. Okay, so here, this slide here, these are the words in the question. The epilimnion is the upper mixed layer. The metalimnion is the zone of rapid change in temperature. And the hypolimnion is where you have the cold water that is dense and isolated. And of course, this is where all the action's going on with oxygen and phosphorus release. So the question is, why is there sometimes higher concentration of chlorophyll, which, is the indicator of plankton growth in the metalimnion. Well, it depends on the light tolerance sometimes of these particular organisms. So if, it may be too much light, and this is something that Julie knows a lot about. Their photosynthetic pigment mixture might be better adapted to light at a lower wavelengths. So sometimes you'll see that. Sometimes you'll see an accumulation also at the top of the metalimnion. And I think that has to do with just the physics of settling. And then, of course, in the hypolimnion, there's low chlorophyll because there's not a sunlit layer. So the plants that do get down there are typically dead. So does Julie, you want to pop in on that?
Yeah, just to say that it is really interesting when we get the chlorophyll data back and we find that it is not the greatest concentration at at half a meter. It's not the greatest concentration, sometimes even at 9 meters. It's the greatest probably right about wherever that metalimnion is. And my explanation in simple terms is that that's where there is more nutrients. I mean, the cyanobacteria can photosynthesize much lower in the water column, back to that good old term. But also, so there's nutrients available. So they pick up the nutrients at night, then they buoy themselves up and they go up to the surface to get more sunlight, and then they drop back down. So it would be really interesting. We don't have the cyanobacteria data at that level, and I'd be really interested in, we have chlorophyll, they also have chlorophyll, to see how much of that increased chlorophyll and oxygen. I think the question was about oxygen, but if you have the plants there, they're going to be making oxygen. So that's part of that explanation too.
Thank you. Maggie would like to know, can Julie talk more about the practical steps to take when a pond is rated potential for concern? Without testing, how do you know if it is safe to go back into the water? Is it enough to no longer see the blobs of scum?
Well, what I tell most people is going in or not going in, it's a personal choice. These toxins are for humans, they're going to irritate your skin, and if you're a sensitive person, or if you're a person that for any other reason is super sensitive, it's just not a good idea. If you're not sensitive and the water's there, then what is the big concern? The concern is toxicity. That's for children on the beach that put their hands in it. That's for dogs that might be eating because they don't care. And especially for dogs going in, they're going to get it on their fur and they're going to lick it off and they're going to have a concentrated effect. So it's a big problem for dogs, I'd say, because dogs just want to go in. And, it feels bad in the summertime when it's hot to say, okay, dog, you have to stay in the house or you can't go. But to the point of the potential for concern, what we record is also, quote unquote, the biomass. So besides the fact that it showed up on the edge with the scum, then we record the biomass. When you see the biomass is really low, that means there wasn't much of it in the water where we measured. So maybe not a big concern. And after several years now with the toxin values, whenever we get a lot of microcystis, we will send those. We'll find out what the toxin value is. And so we have a relationship between the biomass of the plants and how much toxins in the water. And this national studies have been made that for microcystin, that's just the one. We haven't talked about anatoxin, which is a nerve toxin, and we don't have data enough to say anything. So I don't know that really answers it. If you say potential for concern and you have the access to the data, which has the PC, phycocyanin data, and it's low, then there shouldn't be too much of a concern.
Let's just take one more question.
Okay. The next one would be, it's from Melissa. Is white scum a reason to be concerned or only if it has a green tinge? And what is the significance of biofilm?
Well, that's a lot of questions. White, you know, I want to have a fact sheet on bubbles as well, because that gets pretty close to that. I mean, but white would be dead cells mostly. So they aren't active, so that shouldn't be a problem. Biofilm could be a lot of different things, and we just really have to look at it under a microscope to see what's going on. And maybe, Liz, you've had more experience with biofilms, because they could be a lot of things, not necessarily cyano. And that was the case at Ashumet. It was a case at Pill Lake last year. Until I look at the microscope, I can't tell you. Yep.
Yeah, I don't have anything to add on to that.
Okay. All right. Well, I want to, looking at the clock, and I want to wrap up. I want to thank Liz and Julie for what I thought was a really terrific presentation. I want to thank all of you who have joined the call and joined the webinar. We will post the recording of this webinar on the Falmouth Pond Coalition website. We'll also put a link to it on our Facebook page. We will have copies of the slides available for anyone who wants to use them. And I would also encourage anyone who is not on the mailing list for the Falmouth Pond Coalition, or if you've not joined our Facebook page, these are places where you can access information about programs like this, lots of other resources. And if you're not on the mailing list or member of APCC, I strongly encourage all of you to do that as well. They're just doing terrific work. And so, yeah, I want to just wrap it up, say thank you very much to all of you. And I will try to unmute. I'm going to allow you to unmute yourselves now, if you would like. And we can just hang in for a couple of minutes. And if anyone wants to say anything, feel free to do that.